Thank you, everyone, for coming. My name is Brian Ogilvy. I'm a member of our scientific affairs group at BioIVT. And between previous companies, Xenotech, which was acquired by BioIVT a few years ago, and BioIVT, I've been working in vitro drug-drug interactions and ADME for about 28 years. So I really think this is an exciting time in the field, not only in ADME, but other areas as well, with the FDA's roadmap announcement earlier this year in April. And I would really like to extend a big thank you to not only our speakers for coming, but also the large BioIVT team that has really come together and made this happen for us. So I want to set the stage real quickly. When I say new approach methodologies, we're really meaning a toolbox of human-centric methods. So these could be in vitro, ex vivo, in silico, as well as what some people call in chemico. So these are assays that assess pharmacology, exposure, and safety without exclusive reliance on animal models. And as we know, NIH, for one, is not funding any grants that rely exclusively on animals going forward. But these tools are not necessarily here to fill and replace one test with another. They're there to fill critical knowledge gaps, improve translatability, and accelerate development cycles, expand access, control costs, and address ethical considerations. I think most of us don't want to have to use animals unless we absolutely have to, and there are those cases still. But when it comes to new, it doesn't automatically mean accepted. So I think for us to demonstrate reliability, reproducibility, and well-defined context of use, which is critical for all of these NAMs, we have to look across a number of diverse applications. So, why now? I mentioned the FDA's roadmap, but we know that other countries are also looking into this. If you look in Europe, they've been looking at the 4Rs for years. They've already accepted that for cosmetics. We're making some strides here in the US as well. But the regulatory environment is certainly signaling readiness for scientifically validated alternatives. And I think it's up to the field to have well-controlled studies, and for that, we'll need high-quality starting materials. And so for the starting materials, I'll just talk quickly about BioIVT's portfolio. So we go from target to clinic. We specialize in control and disease state samples. We have human and animal tissues and preparations, cell and gene therapy products. So you can think immune cells, leukopaks, and so on, blood and other biofluids, and of course, ADME products and contract research services, which is where I have been working for the last my entire career at this point. And I think we're strongly positioned to support biospecimen requirements for any stage from target to clinic, and especially when it comes to development of NAMs going forward. So I'll talk a little bit about what we bring into this conversation, not as a commercial, but as a provider for high quality starting materials. So for instance, models that are being developed, we have biospecimens, we have high quality, deeply characterized tissues and biofluids. We also have global sourcing under IRB and local regulatory approvals. And we have a large inventory that supports both prospective and remnant collections, match sets, longitudinal data. When it comes to the microphysiological systems, we've got a number of human cell systems. So many of you may be familiar with Hepatopac, and you'll hear more about that from one of our speakers later on, Carissa Cartier. And then we've got also different tissue panels and a suite of ADME-Tox products and services that can supply these offerings that are paired well with development of NAMs going forward. And all of these are coupled with regulatory grade documentation and quality systems. So for in vitro, I'll mention some of the models that I've already discussed. To some extent, we have cultured cells and tissues to assess biological responses to compounds and pharmaceuticals, including chemical and biological entities. And some common systems are 2D cell cultures, as well as 3D spheroids and organoids. And they're often physiologically relevant structure for organ-on-a-chip models, as well as microengineered systems that mimic organ level function and provide dynamic studies of toxicity, pharmacokinetics, and so on. For ex vivo, we have ex vivo NAMs that combine the use of living tissues with the outside the non-animal methods. And this advantage of ex vivo models lies in the fact that there are controlled conditions at all times. So you have minimum alteration and variation in experimental methods. And then many of these feed into the in silico models. So it could be computational approaches simulating biological responses. These are things that we don't necessarily do ourselves, but we're providing services that feed into those. So even if you think of the ADME, we've got all the ADME services that we perform. They go into the physiological-based pharmacokinetic models and other novel AI methods that can be used. And then I want to highlight collaborative validation efforts because they matter to adoption. So one example is a 3Rs aligned liver microphysiological system initiative focused on detecting drug interaction injury. So the design is intentionally regulatory facing. The test sets of unknown hepatotoxicants and controls across multiple commercially available MPS platforms, including HepatoPack, with harmonized protocols and cross-platform analytics that reveal signal versus noise. And these should help provide a path forward in qualification. In this case, our participation in the FDA's ISTAN program will help establish specific contexts of use. And finally, just to talk about a little bit more our clinical collections, we're committed to providing, what you need when you're developing these methods. And beyond that, we operate with over 40 IRB approved protocols, really granting us the ability to offer a wide range of collection types, supporting data that meet global research requirements. And we maintain extensive inventory of over 800,000 biospecimens and have capabilities for both prospective and remnant, like I mentioned before. And we have normal controls, matched sets from individual donors. We have vaccination studies, expertise in blood processing, cell isolations, and we offer customized collection kits to meet specific needs. We also provide comprehensive supporting data types, which can include disease severity, diagnostics, and so on. And then just to end up, we have the global access, like I mentioned before, different types of specimens going across different use cases that we can provide across multiple different countries. And we do try to ensure quality in everything we do. So the quality, ethics, and compliance are absolutely important at BioIVT. We ensure that all biospecimens are ethically procured and our collection and sites adhere to IRB regulated protocols, as well as local and regional regulations and laws. And we do pride ourselves on a superior track record for regulatory inspections, including those by the FDA. And our operations are led by a team of experts specifically dedicated to navigating these regulatory requirements. And we are data privacy compliant, adhering to regulations such as GDPR and the CCPA. We hold several important accreditations, including our clinical biospecimen facility. We have a College of American Pathologists accredited and authorized economic operator status, specifically in the EU. We're also ISO 9001 compliant, and we are Human Tissue Authority licensed in the UK. So just to give you a snapshot that our collections are handled with the highest standards of regulatory approval. And with that, I have a few resources here you can look at later. Everyone will have access to these slides. And I think we're right at time, so I will hand it over to Rhiannon. And so just to give you a quick introduction to Rhiannon, she's scientific director in discovery toxicology at Bristol Myers Squibb in San Diego. She oversees